Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 159th seminar, and I call it a University of Michigan Day. I'm glad to inform you of the second SynBYSS conference that will be held on June 8 to 11, 2026 in Barcelona, Spain. Professor Marc Guer uh, in Spain will be hosting us for this conference, and all of you are welcome to attend it. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Maciek Antoniewicz. He is a professor at the University of Michigan, and I have known him for many, many years. He is a pioneer of metabolic flux analysis and metabolic modeling, and recently he has contributed to microbial consortium research. Maciek, thank you so much for your contribution to the scientific society, and the virtual podium is all yours now. Thank you again.
Okay, thank you. So I have 5 minutes, right?
Yep.
Okay, share my screen. See my presentation?
Yep.
All right. Okay, well, thank you for inviting me as a pioneer for the seminar series. Yeah, it's a great honor to be called that. So I'm going to spend 5 to 10 minutes talking briefly about what my research has been focusing on over the last 25 years. So I first joined Greg Stephanopoulos' lab in 1999 when he published this paper in the Metabolic Engineering Journal. This is actually the very first paper published in the Metabolic Engineering Journal. It's edition 1, page 1 through 11, and it's titled Metabolic Flux of Metabolic Engineering. And one of the quotes from the paper is sort of exemplifies what he believed then to be the core of metabolic engineering as a field. So he quoted the combination of analytical methods to quantify fluxes and their control with molecular biological techniques is the essence of metabolic engineering. So I was really inspired by this paper and decided to dedicate my career quantifying metabolic flux, developing techniques, be able to gain insight into how biological systems take up nutrients and use various metabolic pathways to extract energy and electrons from those nutrients, and then use that for purposes like cell growth. And in metabolic engineering, we're interested in those precursors for generating various products that could be sold. So when I entered the flux analysis field, it was still very much a field that was under development. So the mathematical framework was still not fully established, and there were still a lot of questions whether or not the techniques that we were using were accurate or actually providing an accurate depiction of metabolic fluxes. Since then, over the last 25 years, the field has really matured into a field where we feel very confident in our ability to quantify fluxes. So, broadly speaking, there are three ways that we can use to gain insight into the metabolic phenotype of an organism. Those are flux balance analysis, really pioneered by people like Terry Papasakis and Bernard Paulson. Then there's metabolic flux analysis without stable isotope tracers and C13 metabolic flux analysis with the use of stable isotope tracers. So my research has focused on the third technique, the C13 metabolic flux analysis, um, which allows us to quantify metabolic flux purely based on experimental data. So this is in contrast to techniques such as flux balance analysis, where you have to make certain assumptions about optimality of biological systems, be able to extract some information about what the potential flux phenotypes would be. So FBA has a lot of very useful applications, especially if you want to sample the potential of different biological systems for what they can produce. But it's really this technique of C13 MFA that allows us to gain insight what is actually happening in these systems. So the reason why we're able to do C13 flux analysis is because of a lot of work that came before I joined the field. So it relies on having very detailed understanding of metabolic pathways that was established in the first half of the last century. And then after that, there were dramatic improvement in the analytical techniques, such as NMR and gas chromatography mass spectrometry, which allows us to measure stable isotope traces in a way that's very quantitative that we can use for modeling purposes. And finally, the mathematical framework for C13 flux analysis was established by pioneers like Greg Stephanopoulos himself, Schmidt, and Bichert made very fundamental contributions to the modeling framework of how flux is estimated. And currently, the best technique for estimating flux biological systems is based on this elementary metabolite unit framework that I developed in Greg Stephanopoulos' lab when I was grad student there. So there's a long history that came before we were actually able to do the flux calculation. Currently, as I mentioned, the field is very well established. So in 2019, we published this paper in Nature Protocols titled High Resolution Flux Analysis, where we outlined all of the current best practices in how you can quantify fluxes using the best experimental, analytic, and modeling frameworks. And there we outline in a lot of detail how you can quantify fluxes with the precision and accuracy of about 2% if you apply all of the best technologies that are currently available. So over the span of 20 years, we went from technology that was still very uncertain to what extent you could trust it, technology that arguably has one of the best resolution in of quantifying what is happening in a biological system that we currently have available. So high resolution C13 MFA requires quite a bit of work still. So I would say if you want to do this on a regular basis, it will take you a good year to become proficient in applying this technology. So it still requires quite a bit of modeling work that needs to be that you need to be familiar with. The experimental part of the performing flux analysis requires you to stable isotope traces and significant investment in our analytical techniques like gas chromatography, mass spectrometry. And then you need to be able to use the current tools that are available for performing flux. So in my group, I collaborate with a lot of researchers that want to apply this technique on a one-off basis. So if you're really interested in applying this technique and you want to do it a couple of times, I would highly recommend you contact one of the couple of expert research labs around the world, like myself, but there are also several others who make this as sort of a core priority in their research. over the years, I've sort of demonstrated the application of C13 flux analysis in a wide range of biological systems, ranging from simple microbes like E. coli, yeast, Clostridia, thermophiles, and algae. And in the last decade, we've moved on to more complex eukaryotic systems, including cancer cells, embryonic cells, T cells, and most recently, Chinese cancer ovary cells. And I've listed some of our key publications area. So now that C13 flux analysis field has been established, we are looking forward to how we can further extend this technology, make it applicable to more realistic systems. So we're looking at systems that are dynamic, which original C13 analysis framework didn't allow us to do. System had to be at metabolic steady state. Currently, we're able to extend it to biological system where the fluxes are varying in time. So this allows us to, for example, study autotrophic metabolism. So this is where you have organisms like cyanobacteria or algae that grow on C13 labeled carbon dioxide. We can use more advanced technique of flux analysis called C13 non-stationary flux analysis, which extract flux. Jamie Young is one of the pioneers in the field and currently at the Vanderbilt. We're now also able to look at systems that are non-growing. So by development of techniques that allow us to sample intracellular metabolite pools, we can now also study systems not only in growth phase, but also in a non-growth phase. And finally, what my lab has been focusing on over the last decade is to further extend the technique looking at microbial community. So rather than studying a single organism in isolation, we're now developing techniques called co-culture C13 metabolic flux analysis to allow us to measure not only fluxes in individual microbes, but also identify cross-feeding interaction, identify which molecules exchange and specific rate at which the exchange is occurring. So just to close out, if you're really interested in learning more about our technologies, here are some of the sort of high-level review articles that are published for the last couple of years talking about different methods of C13 flux analysis with application cancer biology, metabolic engineering, and microbiome research. So I don't want to take too much of your time. We have a wonderful junior person here that's going to give us a really nice presentation. So thank you so much for your attention. And if you have any questions, my email is at the bottom of this slide. So thank you.
Thank you so much. This is wonderful. I do believe, this, metabolic flux analysis work is, core of, course, the metabolic engineering, also huge contribution by chemical engineers to biochemical, systems. So I do use, of course, with help from the expert like you. We also published some paper about the flux analysis before. And thank you so much for your contribution to the community. This is basically making metabolism more, clear to understand. And then I do understand that is very important for all the metabolic engineer and biochemical engineers. Thank you.
Thank you.
Okay. Now, the main speaker of today with a longer introduction, Dr. Anush Chiappino studied chemical engineering at the Complutense University of Madrid in Spain. She then obtained her PhD in chemical engineering from the École Polytechnique Fédérale de Lausanne, or EPFL, Switzerland. During her PhD, Anush modeled the metabolism of malaria parasite and identified essential metabolic pathways for the survival of Plasmodium parasite in their liver development. In 2019, Anush joined the laboratory of George Church at Harvard Medical School and the Wyss Institute as a Swiss National Science Foundation postdoctoral fellow. She is collaborating with Professor Stephanopoulos. I mean, that is also advisor of Maciek, and also Basile at MIT. She works at the interface of synthetic biology and computational biology, decoding and expanding genome functions that introduce new chemistries in cells and enhance their safety. She has served as a consultant for biosafety to OpenAI. She has received numerous accolades, including the Excellence Teaching Award by EPFL, annual Jeffrey Hubbard and Melody Schwartz Young Bioengineer Award, MIT Chemie Rising Star, AICH Mentorship Excellence Award, Genetics Society of America de Lille, Nassar, and also Princeton Bioengineering Rising Star. Anush, thanks so much for your time today. Please take it away. All yours now. Thank you.
Thank you so much for the kind introduction, Professor Moon, and I'm very excited to be here. First, let me share my my slides. All right, can you see my slides?
Yep.
All right, great. So I'm very excited to talk today about this project that involves preventing escape and malfunction of cells with an engineered genetic code. So let me start just by outlining what I'll be talking about. So first, the goal that of this project, which is we want to make cells as technologies more safer for use. The problem that we are having is that cells generally tend to exchange genetic materials with the surroundings, with other cells. So beside general strategy suggested before, engineering the genetic code, I'm providing an alternative, an extra solution, and what are the implications of the discoveries I will be talking about today. And this is work that is now as a preprint in bioRxiv, and it's currently also under revisions. So yes, so cells are wonderful biochemical platform technologies, as Professor Maciek introduced before. They have the metabolism with thousands of reactions that sustain life. We find drug targets in metabolism. I am representing metabolism here with this metabolic network. We can produce almost an infinite amount of compounds and peptides that are very interesting. And I'm very interested in producing molecules like non-standard amino acids that can enhance the capabilities of peptides and proteins. But if we want to engineer metabolism, we need to know how to engineer the genomes. So during my PhD, I worked all computationally with metabolism, and then for my postdoc, I joined the lab of George Church to learn how to engineer genomes to expand the biochemical capabilities of cells, and also to make cells safer for use as technologies. So this will be the topic that I will be focusing on today. And now, why do we need cells that are safer for use? First of all, we are having engineered cells and organisms more and more frequently in our daily lives. Like here I'm showing a picture of my bioluminescent petunia. So this has been genetically engineered. So if we want to use cells as technologies, we need them to be to have some safety. We want them to grow in a controlled fashion, to function in a controlled fashion in the desired place and in the conditions. And this could be all the way from a reactor in a very like well-defined space, but we could also use cells for bioremediation in an open space or as a therapeutic. Like we could consume it as a probiotic. And in those open conditions, even in a culture, these cells may be exposed to other cells, like Professor Maciek mentioned before. He's working in co-culture conditions. And we wouldn't want our cells to behave like, we wouldn't want our cells to lose their function, the one that we engineer, and we would lose control of our technologies. And this could happen due to natural processes, like, as I was mentioning before, the exchange of DNA from one cell to another, but also due to natural processes, due to negligence or misuse. So in order to make cells safer for use, for around 20 years, they're in the field of cellular engineering. They're having like this concept of biocontainment. So we are trying to make cells grow only in the conditions where we want them to grow. And the tightest biocontainment approach that has been suggested today is one that was published actually by the Church Lab, where there was 1 in 10 to the 12 cells that were escaping only. And this was because these cells were made dependent on a synthetic molecule that is a non-standard amino acid. And what is a non-standard amino acid? So we all know that there are 20 standard amino acids. These have amino carboxyl groups highlighted here, and these allow the amino acids to form peptide bonds. And in this case, I'm showing phenylalanine. Well, non-standard amino acids have additional functional groups beyond the 20 standard amino acids. And like in this case, this has an extra phenyl group, and this molecule is called biphenylalanine or BPA, which is the one I was showing in the previous slide. And there are hundreds of non-standard amino acids. There are over 400 that have been shown to be incorporated into proteins, but theoretically, there could be almost an infinite number of non-standard amino acids. And these amino acids could have new structures with the same type of atoms and bonds as standard amino acids, but they could also introduce new atoms, like as we see here, compared to the standard amine, or new types of bonds. So non-standard amino acids have been, like, the interest in working with these amino acids has been increasing, primarily due to their use in this very special peptide, semi-glutate derivatives. The Ozempic drug that has a huge market value nowadays actually uses 2 non-standard amino acids. This is a drug that is used to treat diabetes and obesity. And these 2 non-standard amino acids provide the properties that have made this derivative of the semi-glutate so successful. So one of the non-standard amino acids enhances the binding, and the other one enhances the stability. So it lasts now for over a week in the blood. So there is an increasing interest in understanding how we can produce and use this type of amino acids. Now, going back to how we make cells safer for use, besides biocontaining by dependency, we could also think of strategies that bioisolate cells, generating what we call a genetic firewall, so that cells would not accept any external DNA. And how can we do actually both of these? How can we take both concepts and work with both of them? And so that we can have cells that are safer for use, but also enable new amino acids. So enable this capability of using cells as biochemical platform technologies for expanding the proteins with new amino acids. And we do this by engineering the genetic code. So just a brief introduction of what is the genetic code. We know how to go from DNA to proteins through transcription and translation. Well, the genetic code are these rules that, like, defined with 3 letters how the DNA codes for information of the amino acids. And this, the coding of the DNA information to the protein information is done through a very important molecule that is called the tRNA. And it happens through the recognition of the codons at the mRNA level with the anticodons in the tRNA. So the problem, or it's not really a problem, it's actually a feature, is that there are 64 codons. There are 64 combinations of 3 letters. At the DNA level, we have 4 letters. So there are 3 letters that define a codon within a gene, but there are 20 standard amino acids. So there is some degeneracy in the genetic code that determines that we end up having, as I'm showing in this table, higher number of codons defining what amino acid is being encoded in those positions. So in this case, I'm showing the codons that are linked to serine. So we have TCG, TCA, all at the DNA level. This all code for serine. And here I'm showing also what are the tRNAs that are linked in E. coli to decode those codons and the corresponding anticodon and gene. So these interactions between codons, anticodons are pretty well known in E. coli, at least. And it's just important to remember this information for the rest of the talk. The fact that we have well-known interactions between codons and anticodons in E. coli. So how do we engineer the genetic code? Since I said this is the best strategy to move forward with bioisolation and biocontainment, what we do is we select a set of codons from the genome, most commonly the least frequent ones. Let's say in this case, the TCG codon, coding for serine. We would synthesize a genome that doesn't have this codon, but we substitute this codon by a synonymous one so that the proteins remain the same, so that all those positions still code for serine, but the genome wouldn't have this codon anymore. And once that genome is complete, then the corresponding tRNA that decodes that codon is not needed anymore, is not essential. And therefore, we can also delete the gene that codes for that tRNA. So the cell, in theory, contains all proteins that are not perturbed. They contain the same amino acids, but it lacks codons throughout the genome. And what do we do with those cells? We repurpose those codons. So we reintroduce those codons in any protein of interest to be able to incorporate with an orthogonal translation machinery, like an orthogonal tRNA and an orthogonal enzyme aminoacylating that tRNA. This is called an amino acid tRNA synthetase, a new protein that contains the non-standard amino acid. And this engineering these enzymes is not that straightforward. We need to figure out how to engineer the catalytic pocket, and also it could be needed to engineer the anticodon position, depending on what codon we want to introduce a non-standard amino acid. So as you see, engineering the genetic code not only ends up giving us an incomplete translation machinery with a lack of tRNAs, but also allows the incorporation of new amino acids. So the fact that we have an incomplete translation machinery means that these cells, in theory, cannot read extracellular DNA that relies on 64 codons. Because these tRNAs are not present, the cells would not be able to read, our cells would not be able to read all the complete set of 64 codons for the natural serine amino acid. Now, this has been done before. Like the first genomically recoded organism that was created was in the Church Lab. It was published in 2013 by La Joye et al. And this cell lacks the stop codon TAG. And then it was substituted by any of the synonymous like TGA and TAA. And then the release factor was also deleted in this cell. So then these cells can very easily incorporate non-standard amino acids in the stop codon, which is also the codon mostly used in genetic code expansion for non-standard amino acid incorporation. And now the Second cell that has been published that is also genomically recoded has 61 codons. So it has 3 codons that are free. This was published by the Jason Chin lab in the UK. And the first strain with the synthetic genome was published in 2019, and with the tRNAs, 2 extra tRNAs deleted and the release factor deleted was published in 2021. So beyond recoding the TAG codon, they deleted these 2 tRNAs from the genome. They substituted it by these synonymous codons. So again, all the proteins still contain the serine in the right place, but then they deleted the corresponding tRNAs. And just for the rest of the talk, we can, I will be coupling these 2 codons, TCG and TCA, as TCR. And if you could remember that we have TCR codons linked to 2 tRNAs that were deleted, that's enough to understand the rest of the concepts. And there will be another cell that has 57 codons that is being developed in the Church Lab, and for which we have put out an update. It's in bioRxiv as well. All right, so I mentioned that these cells are safe for use because they lack tRNAs. And I am now asking this question. Is the 61 codon E. coli without 2 serine tRNAs bioisolated? Like, is this genetic firewall really as we thought? So previous work showed that if DNA that is extracellular contains those serine tRNAs, those 2 serine tRNAs that were deleted in the cells, the bioisolation is broken. But the question is, if this DNA doesn't contain the tRNAs, this cell should be bioisolated because it doesn't have those 2 serine tRNAs that recognize the TCR codons. I challenged that, and I did that by providing to these cells antibiotic like plasmids that contain antibiotic-resistant genes with TCR codons. As we said, these cells don't have the tRNAs to recognize those TCR codons. So I was expecting that without these plasmids provided and with these plasmids, the escape rate would be like the same, which is no colonies at all, no cells when we expose them to antibiotic. But what I saw was that these cells were surviving, the ones that contain the plasmids, the antibiotic-resistant genes with the TCR codons that are in theory not eligible. And they were surviving in this specific example at 5 orders of magnitude higher than expected, which is you would expect in the acquisition of antibiotic resistant 1 in 10 to the 9 cells would acquire antibiotic resistance based on spontaneous genomic mutation. So this was a bit mind-blowing for me at the time. I thought, what is happening? I started doing a lot of analysis. My first, thoughts were like, I have a contamination. I need to do deep genome sequencing. I did deep genome sequencing. The cells were what my cells, SYN61 delta 3, without the 2 tRNAs. I confirmed that the 2 tRNAs were deleted from the genome. Then I thought the plasmids have mutated. They probably shifted from the TCR codons that are not legible to something that is legible to be able to produce the enzymes. The plasmids were not mutated. They still contain the TCR codons. And I'm going to show you the results that we see based on proteomics and some additional analysis I did. And I want to say that I did expand this analysis to 5 different antibiotics, more than 20 different plasmid designs with different gene designs. And I analyzed over 100 number of escapees individually with all these data sets. So it was really a generalized phenomenon. So what I first saw that was also really mind-blowing was that in those escapees from carbenicillin, the enzyme was detected. So, and here I'm showing the gene And all these positions that are highlighted in black are all serine positions, but the blue ones are the positions where serine is coded by the TCR codons. So with proteomics, and here I'm mapping the peptides that are detected that detect the protein based on proteomics. What we see is that with all peptides mapped throughout the protein is that the whole protein was detected in these cells, meaning that the cells are able to produce these proteins despite not having the wild type, the 2 wild type tRNAs that decode TCR codon. So that was the first thing that didn't allow me to sleep for days from excitement. I mean, worry as well, because we don't want this to happen, but it's also exciting to see such discoveries. So then the next question is, what amino acids are being incorporated in those positions? So going back to the analysis and seeing what are the peptides mapped to those positions, we can identify what are the amino acids that are being incorporated. And I saw that there were serine, proline, serine, arginine, but then serine was incorporated at more than 98% of the cases. So there is a high bias for serine incorporation. However, my question was like, it might be that all amino acids are being incorporated, but we are only seeing serine because these are natural positions for serine. So these enzymes might only be functional when serine is incorporated. So it might be just a survivorship bias. So in order to answer that question, I introduced in the same plasmids where I had these antibiotics with TCR codons, I introduced another gene that's a reporter. And this was a GFP reporter that contained 5 instances of serine coded by TCR codons. So I again saw the escapees, and when I analyzed, when we did the proteomics, we detected both the antibiotic-resistant enzymes, but also the reporter was, as you see with all the peptides mapped throughout the protein, the full protein was detected. And to understand what amino acids are incorporated in those TCR codons, I checked through these peptides, and I saw that serine is incorporated in more than like in 92%, at least 92% of the cases, which suggests that there is a new mechanism that enables these cells to incorporate back serine into TCR codons. So we did tRNA-seq analysis, and we are now currently collaborating with the Kleiner lab in Princeton to do LCMS of the tRNAs. And this is our current hypothesis, and I will explain what the tRNA-seq analysis said. So our hypothesis is that when we provide these plasmids with TCR-containing antibiotic-resistant genes, and we expose these cells to antibiotics, so we are selecting. There is a stage, these cells escape because there are 2 stages. And one is that there is mistranslation. So these cells are able to produce, to some extent, some functional proteins, although they are not incorporating to the full extent the right amino acid. And then upon continuous selection, we are mutating or modifying available serine tRNAs that in theory were not able to interact with those TCR codons, so that they are able to recognize back those codons. And basically what we saw with our tRNA-seq data is that there was a mutation that would enable, a single point mutation would enable this tRNA that is available in the cells, would bring back this anticodon and therefore the capability to recognize back those TCR codons. And here I'm showing some sequencing data for that SER W or X tRNA. And yes, so we rediscovered that life found a way through this new approach or mechanism. And just to prove that this type of mutant tRNAs, because these two tRNAs are around 60% similar in sequence. So just to check whether this mutant tRNA is functional, we went and checked in vitro whether this sequence with these two anticodons would be able to be aminoacylated. So we run this reaction in vitro where we have the mutant tRNA, the enzyme that is aminoacylating it with serine. We allow this reaction to happen so that aminoacylation happens or not, depending. If the mutant tRNA is not functional, this wouldn't happen. And then we add an enzyme, an RNase, that will cut this acceptor arm so that we can detect through LC-MS whether the amino acid serine is present or not. And what we saw is that aminoacylation levels of these mutant tRNAs was almost to the same extent as than wild-type tRNAs, which validates the fact that these mutant tRNAs are recognized by the corresponding enzyme and therefore are functional. So to summarize this discovery, we have shown that although DNA, external DNA, doesn't contain TCR codons, it could be it could break the bioisolation of these cells. And this happens when we, and by the way, I also studied two different numbers of TCR codons. This happens when these antibiotic resistant genes have up to 5 TCR codons. Beyond 5, I haven't seen escapees. But within that window, we see this problem with the breaking of bioisolation. And this happens through mistranslation and then modifications of mutations of the tRNAs. And as you can imagine, if we want to use cells as biochemical platform technologies to produce non-standard amino acids or peptides or proteins that contain non-standard amino acids, and we start having mixtures of tRNAs, then the production yields will be reduced because we are not only incorporating non-standard amino acid in those positions, but also we are incorporating the standard amino acid back, which in this case is serine. So the question is, how do we solve this problem? So I was trying to think of a solution and thought, what about using kill switches? So we know kill switches are toxic proteins. They are that when they are expressed without any error, they will kill the cell. And when they are not expressed or when they are expressed with errors, the toxin will be inactive and the cell will survive. And what we are trying to detect here is incorporation of serine into TCR codons. So what I did is I identified what are essential serine positions using protein language models. And what I call essential here is positions where a machine learning model for proteins, these are the protein language models, will tell you it is highly likely based on evolution that serine is the most likely amino acid to give a functional protein. And I identified various of these positions to minimize the probability of escape. And what I did is I coded multiple serine positions with TCR codons of those most essential positions, so that when there is a serine tRNA that is recognizing TCR codons, the toxin will be active, the cell will die, so serine is incorporated. And when there is no tRNA or when there is an tRNA that incorporates a non-standard amino acid, the toxin is inactive and therefore the cell survives. And just to show you how the best of my I call it genetico-sensitive kill switch works. This is the number of escapees I was seeing in my plate upon electroporation of the plasmid with a kanamycin resistant gene with 4 TCR codons. And this is with the same antibiotic resistant gene, this is the number of escapees I see when the cells have my best kill switch after 10 days. So the kill switch reduces 4 orders of magnitude the escape rate compared to this genetic code engineered cell. And overall, the escape rate is compared to any wild type cell is reduced 8 orders of magnitude. So we have 1 in 10 to the 8 cells that are escaping, acquiring antibiotic resistance when we have a kill switch. So what I think are the contributions of this project is, one, from the basic biology point of view, we have shown that these cells are not as safe as we expected, that even if we have a heavily engineered genetic code, thousands of mutations throughout the genome to prevent the cells to recognize wild-type genetic codes, there is still a narrow, but still there is a window where they can escape, so they are not totally isolated. This can happen through mistranslation and to what I call a dynamic tRNA pool. So tRNAs that initially could not interact with other codons, they are by selection being able to acquire this capability, and therefore we are enabling evolution of an engineered genetic code. And what this work enables too is new technology. So we are having safer cells. I will not say these are unescapable, of course. As we can never say that cells will not escape at all in any scenario. At least we should be cautious with that statement. So we are reducing 8 orders of magnitude the acquisition of and spread of antibiotic resistance compared to wild type. And we are also allowing, because all these codons remain free for use, the incorporation of multiple non-standard amino acids, and with higher fidelity because we are preventing the incorporation of serine again. So ultimately, what the idea is to enable more tighter and tighter biocontainment is to suggest layers. So we suggest, we can recode the genome. This provides a first layer of biocontainment. Then we can introduce genetically sensitive kill switches and reporters so that we prevent these more mutations of modifications of tRNA. And if those happen, we still have a reporter that will tell us whether those cells have escaped through this mechanism. And then the idea is to repurpose as soon as possible those codons to new amino acids. And if those amino acids are in essential genes, as it was proposed with BPA before, this was proposed with the TAG codon, but we could do this with sense codons as well. The higher number of proteins, essential proteins that incorporate those non-standard amino acids, the higher level of biocontainment, which basically is like we could have a new genetic code with 21 amino acids or more. And the idea is this way we could have safer technologies with codons reassigned for the different applications that we would like. So with that, I would like to acknowledge George for supporting my postdoc, all our collaborators. Bogdan has been leading the proteomics work. My other colleagues have been helping with different experiments in the project. Felix did the in vitro tRNA charging, and different collaborators have been helping with revisions for the paper too. GenScript for DNA synthesis. As I said, in Princeton, we are currently working with LCMS of tRNAs. And the team at HMS and Church Lab, funding sources, and thank you all for your attention.
Fantastic. So this is amazing work. I probably biased because my research also heavily involved kill switch or biocontainment. Let me start with a couple of questions. The first question is more technical question. I just wonder, because you show us tRNA mutation, and my guess is the original wild type without mutation, the tRNA, that also coexist with the mutant. Am I right?
That's what the suspicion I'm seeing. I think is that there is like a reversible reaction that is either modifying or mutating that tRNA from the wild type into this mutant that is enabling the recognition because you still need this wild type to be able to recognize the codons that are
The
available in the
original
genome is
codon.
the most optimal tRNA, but you need this to some extent to recognize that those, TCR codons that are
So
present in lower
that
extent.
is somehow kind of mystery to me because your genome sequence should be just one, but you still have the two different tRNA with the two different, anticodon. And how?
So I am not seeing the mutations. I'm not seeing duplications of the tRNA at this point, and I am also not seeing the mutations at the tRNA level, like in all escapees. I am seeing them in the tRNA-seq data. So I think this is happening post transcriptionally, and this is what we are confirming with our collaborators at Princeton regarding the modifications per se. Also, based on proteomics, relative expression analysis, we are seeing some tRNA modifying enzymes slightly overexpressed. And one of the enzymes is introducing a methylation. And it seems like, based on previous literature, methylations around the anticodon can expand the
Got it.
recognition capability of tRNAs.
That makes sense now because so that happening post-transcriptional
Mhm.
level in that way,
Mhm.
you can have the both of them at the same
Mhm.
time.
Yeah.
That makes sense. My second question is more kind of more like the big picture question, potentially important for you, I mean, for your career. I'm kind of working on biocontainment for now almost 10 years. I do understand this heavily recorded uh gigantic engineered bacteria. I hope I believe those bacteria would have the best uh you know killing efficiency in terms of biocontainment because this is the gigantic recorded. But interestingly, you also show, this is not completely, isolated. That's also interesting data to me. The question is, unfortunately, if I understood or, remember correctly, so far, this gigantic recording only happened in E. coli, right? So how are you gonna generalize this kind of approach to Pseudomonas, to Rhodococcus, some other bacteria because we are not, rich like
Yeah.
the church lab or the Jason Chin's lab, but still we only have the E. coli for the biocontainment, but there are multiple different bacteria that's useful for applications. We need biocontainment. How are we gonna,
Yeah.
you know,
So
address this
I
problem?
think there are different things. So first, it is to say that we are having trouble with 7 codons. Like it seems to be a lot of codons to make a viable cell. So it is wiser, based on this experience, if we go to other organisms that are even, less known than E. coli, to work with a lower number of codons to be recoded. So that's the first thing. The other thing is that we have also seen that we expected that because we are not changing proteins, this won't affect so much the fitness of the cells, but coding regions are overlapping with regulatory regions. So we are perturbing promoters, ribosome binding sites throughout the whole genome, and this is reducing fitness a lot. So I think I am also a computational person, so I'm a bit biased, but I think focusing more on machine learning approaches for genome design is key for us to be able to design faster, better genomes. And this is applicable not only to E. coli, but to other organisms. And then we also need faster ways to synthesize large genomes, and hopefully
Yeah,
we'll get
fantastic. I want to see more of recorded genome in other non-model organism as well. In that way, other researcher like me also benefit from those things because I used to be E. coli guy, but when I start my own lab, I start to move toward more non-model organism like, Pseudomonas, Rhodococcus or some other, one, because I realized that it's more useful for the real world application later on.
Yeah.
Yeah.
But I
Fantastic.
think we are working toward those goals.
Oh, you
So
are working toward that one?
I mean, we are trying to generate, one of the goals of my future lab is to be able to have like better machine learning approaches for genome design. So in a way, we are trying to enable those tools to be available for everyone.
Good. That's fantastic. Let me check whether we have question from audience. Okay, we don't have, and also we are a bit over the targeted hour. That's now 11:04 a.m. Central Time. So let me close and then we just chat afterward. Okay, so thank you all for joining and staying today. We'll meet again on April 24, Thursday, the same time, the same Zoom link. We'll have Professor Kelvin Lee at the University of Delaware and Dr. Hao-Sheng Shen at National University of Singapore. I believe very inconvenient time zone for him, I believe. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. 1 second.